For most of human history, our understanding of biology was strictly bound by the limits of our own eyes. We could study bones, organs, and leaves, but the actual architecture of life remained hidden in a dimension too small to perceive. That physical barrier finally broke in the 17th century with the invention of the microscope. By arranging glass lenses to carefully bend light, early researchers could suddenly magnify microscopic matter. Early pioneers relied on complex, handcrafted instruments to illuminate their samples. Looking at this specific 17th century apparatus, you can see how a glass globe filled with water was used to focus the light from a flame directly onto a tiny specimen beneath the lens. With these tools, human sight was no longer the definitive boundary of biological reality. An entirely new frontier of microscopic life was instantly open for exploration. In 1665, an English polymath named Robert Hooke pointed his compound microscope at a thin sliver of cork. He sketched a rigid pattern of compartments. Reminded of sparse monks' rooms, Hooke called them cells. But this was a biological ghost town. The cork was dead. The living matter dried up. He observed only the empty cellulose walls left by the plant. A more active discovery came shortly after from a Dutch draper named Anton van Leeuwenhoek. Using superior single-lens microscopes with much higher magnification, he observed frantic, translucent organisms swimming wildly in drops of water. He called these living, moving entities animalcules. These initial glimpses proved that microscopic structures existed, yet they remained isolated curiosities. Science had a fragmented list of tiny objects, but no one suspected these cells represented a universal rule for all living things. It took nearly 200 years to connect those fragments. In 1838, a German botanist named Matthias Schleiden examined a massive variety of plant tissues. He realized that despite their differences in shape and function, every single plant structure was built from the exact same common unit, the cell. The very next year, a physiologist named Theodore Schwann conducted parallel investigations into animal tissues, looking for similar structural rules. Their combined insights formed two pillars of cell theory. All organisms are composed of cells, the basic unit of structure. Though plant cells have rigid walls and animal cells have flexible membranes, their internal machinery operates on the exact same structural blueprint. By merging their findings, Schleiden and Schwann erased the structural boundary dividing the natural world. Plants and animals were no longer entirely separate categories of matter. They were simply variations of the same cellular architecture. However, this initial theory had a massive logical gap. It successfully categorized the building blocks of life, but it failed completely to explain how a new cell actually came into existence. For centuries, the prevailing assumption was abiogenesis, or spontaneous generation. This was the deeply entrenched belief that living organisms could simply spring forth from decaying or non-living matter, like maggots spontaneously appearing on spoiled meat. Disproving this ancient dogma required rigorous, highly controlled laboratory experimentation. Scientists like Louis Pasteur had to design meticulous tests, like his famous swan neck flask experiments, to prove that microbial life only appeared when exposed to pre-existing airborne microbes. Until science could replace spontaneous generation with a proven mechanism for cellular creation, cell theory remained an observational categorization system rather than a predictive law. In 1855, German physician Rudolf Virchow provided the mechanism for cellular replication, defining a third tenet, omnis cellula a cellula. All cells arise from pre-existing cells. This principle established an unbroken lineage of biological continuity. A cell never simply manifests out of thin air. It is only generated when an existing cell physically divides to create copies of itself. This principle shifted the search for the cause of disease. If every cell has a cellular ancestor, then diseased tissue must originate from a malfunction in the cells themselves, rather than the bad air or humoral imbalances doctors had blamed for centuries. Cancer is the ultimate illustration of this principle. It is not an external force striking the body, but a disease driven entirely by pre-existing, once normal cells undergoing mutation and dividing uncontrollably. By establishing the principle of cellular continuity, Virchow provided the framework for modern pathology. Doctors could now trace the progression of an illness by observing how specific cells divided and changed over time. As technology advanced, so did the theory. The discovery of DNA as a hereditary blueprint and the mapping of complex metabolic energy flows added deep mechanistic layers to those original 3 tenets. We also found biological edge cases that test the traditional idea of isolated compartments. Skeletal muscle fibers and certain fungi grow into long, continuous tubes filled with freely floating multiple nuclei. But these structures do not break the rules. They are highly specialized derivatives that originally formed from the division or fusion of standard cells, maintaining that unbroken chain of replication. Today, the study of viruses, which require a host cell to replicate, and the push to build synthetic, self-replicating chemical systems in labs continue to test exactly what biological structures count as being alive. Cell theory remains a successful, functional framework. It continues to expand as new data emerges, providing a consistent logic that unites the disparate fields of modern biology.